So you have heard in the first part of the day, in the morning, starting with Noam Harel, that it's now possible using high-field imaging to map even single cell layers and to come up with information with respect to the function of cortical layers and which has not been available in the past. And this brings, of course, neuroimaging in the living human brain much closer to architectonics, microstructural information, and helps to ask new questions. So I'm happy that our next speaker, Rainer Göbel from Maastricht, is presenting high-resolution function imaging and anatomy of the visual cortex. So thank you, Rainer, for coming.
Thanks, Katrin. It's a great pleasure to be here. And in the next 25 minutes, I would like to share with you some of our results and some more general remarks about the organization of the visual cortex. And it's actually here in this introductory slide already visible that if a person looks at objects, which was the task in these images here, you see that many areas become activated, but it's difficult to see structure in there. So whole patches of cortex are activated. And it is important that this can be understood best at multiple scales. For example, at the scale of areas and networks, which is typically done with functional MRI at, say, 3 Tesla, which has shown certain pathways in the visual cortex, which I will show you in a schematic. But I will focus today on high-field imaging, on high-resolution imaging, where we zoom in these kind of areas and will start to understand not only retinotopic organization, as we have heard before in Dr. Glasser's talk, but also zooming into retinotopic cortex and other regions in the visual system to see the coding principles in these areas. Sorry, yes, sorry. So this is actually what I want to present today, so that we have a multi-scale understanding of the visual system, which starts from kind of areas and networks, and then it goes further to zoom into mesoscopic scale structures, which are akin to cortical columns and cortical layers, as Katrin has just alluded to. To do that, I will show you that with ultra-high field imaging, we are now at a verge of seeing new structures in the brain. So it's not just seeing more of the same, but we see qualitatively new information. And that's actually what I want to show you where we are in this field. And then I will show you, when I first look more on the tangential part of the cortex, where we see the functional organization of the cortex, I will then look also, with one example, in the depth recording of the cortex using ultra-high field imaging, so that we can disentangle bottom-up from top-down processing. So first, here's a schematic overview of the most large-scale functional organization of the visual system. You see the pathways which have been identified, of course, in the monkey, but also in the human brain. You see the dorsal pathway and the ventral pathway. The dorsal pathway is known to process spatial information, also attention, sensory motor mapping, orientation in space, functions like that. And the ventral pathway, with many areas which have been mapped in the human brain, is devoted to object recognition, kind of transforming features to invariant representations at higher levels of the visual hierarchy. You also see these bidirectional arrows, which indicate already that visual analysis has a fast feed-forward speed, but also slower and more elaborate backward projections. Actually, there are as many backward projections as feed-forward projections. So the brain is a recurrent processor to make most sense out of the input it receives. And we have also connections to the prefrontal cortex, which helps, of course, to implement higher cognitive functions like visual attention, like visual working memory, and so forth. So all these structures here kind of depict the kind of macroscopic organization of the visual system as identified by anatomical, but also largely by functional experiments. Important point is the areas which are shown in this overview graph here, how they are localized. I showed you before that many areas are active, giving the same stimulation, but you need to basically localize with specific stimuli these specific functional areas. But these areas are not as clear-cut as people might think. That means that these areas do not respond like the face area, not exclusively to faces. It's just that it's the most strong response. So if we do functional localize and show a lot of different kinds of stimuli, visual stimuli, like faces from human, faces from monkeys, body parts from human and monkeys, objects, different sculptures and different stimuli, we get a nice picture of what these specialized areas are really specialized for. So it's important to probe them, not with just one or two stimuli, because you can quickly say they are specialized for something. It's more important to really use many different features. And then, if we use 7 Tesla imaging, we see very fine-grained patterns of activity, like here is the extrastriate body region, which responds, of course, strongest to human bodies, but also strong to monkey bodies, and also to human and monkey faces. So it's not exclusively to one function. If we go further and look in the ventral cortex, like Kevin Boehner has done before, we see indeed also with high field imaging, like 7 Tesla, the kind of separation of FFA into substructures, like we saw the posterior fusiform face region here, which Kevin has explained, and here's the medial fusiform region, here's the OFA. But you see also that with high field imaging, even the sub-area of the medial fusiform falls into subclusters again. So that means we have a hierarchical structure here, and with higher resolution, we get finer and finer structures, even within areas we consider as one region, like the FFA. And that's an important first step to zoom into from a global point of view to a more and more local point of view. Here are some of the typical areas which we can map in individual brains very easily and find precise location of them after, of course, the retiotopic areas, which I haven't shown here. We get what I call mid-level visual areas, which have some specificity, but it's not exclusive. And important point is, and I come back to Kevin Weiner's talk, if we can do this, as Kevin also showed, in post-mortem brains and in vivo brains, if we use these ventral visual areas and use the medial fusiform sulcus to see in individual brains how they are localized, this does not help us much in how to bring this together in an atlas, where we want to have a kind of a global, common representation of the relationship between these areas. And we found by collaborating with Kevin Weiner and others, and Kul Spector and others, that basically if we use for the alignment, a curvature-driven alignment combined with landmark alignment, which is actually used here on the right side, we see that if we include the MFS, the medial fusiform sulcus, anatomically as a landmark to drive the alignment, then we get also in the group data very nice alignment of the medial fusiform and the posterior fusiform face regions. So these face clusters align very well in the group map if we use in our alignment the MFS, an anatomical structure, as a landmark in addition to curvature alignment. And if you want to see the details of this, you can see the poster by Martin Frost here at this meeting. And just to round this up, you see here in the 3 different colors how the normal alignment, the kind of just bringing it at the same cortical space, then using curvature-driven alignment, and finally using the MFS improves the correspondence across subjects. The lower the values there, the better the correspondence across subjects. Then the next level of zooming in from the macroscopic point of view to the fine-grained point of view, the next one is within areas kind of simple features like retinotopy. I call them simple features because they have a gradient across the brain areas, as Dr. Glasser has also shown in the previous talk. One of the previous talks. And there we can, of course, use different techniques. Here I'm using the so-called population receptor field method, but it doesn't matter exactly which we use, to find the kind of foveal peripheral eccentricity gradient here, and here kind of the mapping of horizontal vertical meridians to demarcate the areas, as we have learned in one of the previous talks. So that's the next level, because we see now structure within areas. For example, within V1, dorsal, ventral, and the other areas, we can now see gradients of where we are in the visual field. But even that is not really showing the computational aspects of the visual system. And what I mean by computational aspects, I mean the features which are within specialized brain areas to basically represent and compute information and process information. For example, in the face region, we know if we show a face, the face area or one of these many face areas or all of them become active, but we do not know how a face is coded within the face area. And the quest we are doing in the last years, which we have begun with some other colleagues, is basically to do just that, to zoom into these areas and to resolve informational structures within specialized brain areas. And the idea is that if we have standard 3 Tesla brain imaging, we basically reach a level of this global scale, macro scale, where we see specialized brain areas like the face region or others, but we also see pathways, networks, resting state networks, and things like that. This is what we see if we use conventional brain imaging. If we push the limits and go to ultra-high field imaging in the sub-millimeter range, then we not just see these structures better. The important point I want to make in my talk is the take-home message. If we really increase functional resolution just a bit is very tough to get, but if we do that, we suddenly see new structures, and these are in the tangential cortical space, cortical columnar-like structures, and in the vertical, in the depth space of the cortex, kind of a coarse view on laminar structures. And that allows us to basically interpret imaging data in a novel way, because we learn something about how information is coded within these specialized brain areas. And of course, this goes back to the famous Hubel and Wiesel studies, which showed with electrophysiology that within a patch of cortex, in V1, for example, for this patch, there is a whole set of cortical columns for all orientations for the left and for the right eye, and there are even other structures for color and so on, and they call these structures for the same patch of the visual field a hypercolumn. And so that means it's not just the retinotomy, but in each retinotopic location, there is other features within these retinotopic locations, and these are the things we are interested in, because they reflect what this area is really representing at the feature level. And the studies by Hubel and Wiesel can be best understood to have shown that in V1 we have feature detectors for different orientations at each position in the visual field. And these feature detectors are the computational elements, the alphabet of the brain area. And in optical imaging, we actually see, this is in animal research, very nicely the structures and the colors code the different orientations, and they meet at so-called pinwheels. And these are kind of structures we are interested to see with high-field imaging in the human brain. This is the challenge. How can we achieve that? So the most important point is, if we increase the resolution from a 3 Tesla scanner of about 2 millimeter to a 7 Tesla or higher scanner to sub-millimeter, say half to 1 millimeter resolution, the point is that we would not really see new structures in case that the neurons would be randomly layouted in the structure. Because if we pool now here, we also get just a sum of all orientations. We cannot separate them because we do not resolve single neurons, of course, in this schematic view. But we are lucky that in many brain areas, especially in the visual cortex, the neurons cluster into these cortical columns. And that means that, for example, horizontal neurons are not randomly displayed, but they structure and form functional patches which basically correspond to a structure of horizontal, in this case, oblique and vertical feature detectors. So to find the feature detectors in V1, we do not have to resolve the neurons. It is sufficient to resolve the next higher hierarchical structure, which are the cortical columns. If we resolve this structure, we are able to see the coding principles. Of course, this is only an approximation of the neurons, because within the column, there are some variabilities. There's not exactly the same response in each neuron. That's, of course, a simplification. But at a whole, they are more similar to each other than, say, if you go tangentially here, suddenly you get an abrupt change, and then you find many neurons which code a different orientation. And that's the classical columnar model. And we do not think that we can resolve this completely here, but that we get some information that our voxels become so small at 7T that they are driven mainly by single columns or just a few columns, and not by 10 or 100 like in 3 Tesla scanning. We also hope to see the layers here, because if the cortex is 3 millimeter thick, 2.5 to 4.5, something like that, then we should see with sub-millimeter imaging also the laminal structure, at least in a very coarse way. And that's also something we are interested in. So is that possible? In this seminal paper by Isa Yacoub and colleagues, it was indeed shown that with high field imaging, in this case 7 Tesla, and a certain spin echo sequence, it was indeed possible to resolve orientation columns in V1. So this paper is very important because it shows that we can indeed resolve these very fine-grained columnar structures in the human brain using brain imaging. The price to pay is we have to move on to high-field imaging and to sequences which are less error-prone to draining vein effects. I come to this in a moment. This is just for comparison a monkey optical imaging of orientation preference, and you see the layout, the structure is of course not identical. It's even not identical from one individual to the next. It's quite idiosyncratic, but the general logic is very similar, namely resolving these pinwheel-like structures and the whole set of orientation features in a patch of V1 cortex. The problem with that study of Isa Yacoub was that only one slice was scanned and they looked for people which have a straight calcarine sulcus. Then they could just put one slice in one of the banks, here in the lower bank, and then basically they had a thick slice, so they averaged vertically the columnar information, so no layers here, and they got kind of, in this tangential direction here, the kind of columnar organization. Actually, this piece here is exactly what you see in the previous one here, right? This is basically that piece. It was a bit bigger, but this is in principle like it is. So the problem of this approach is that we cannot see in structures which are curved, because the technique limits that to just one slice of spin echo EPI. So by collaborating with Camille Ugerbil's group, including Isa Yacoub, but also David Feinberg, we kind of used the so-called Grace sequence, which I will show in a moment, to basically circumvent this problem, and we are now able to use a spin echo-like sequence, but with the possibility to have multiple slices so that we can also image in depths and curved cortex. That's very important, this technical advance. And of course, we also need to image the layers to some extent at least, because we know from Keiichi Tanaka's work that the columnar concept is of course simplified. It is not the case that a column goes straight vertically through without change. There are sometimes invasions from other features, the structure shrinks or expands and so on. So we need to really image both dimensions, not just one slice, and that's what we have achieved also by methodological advancements, in the sense that we basically invented some tools. Does not proceed. Oh, thank you. In these tools, we basically allow to model the depths of the cortex and the tangential direction where we expect the columnar features with so-called 2D grids, which we move into the cortex at different depth levels, and we want to sample then the functional data from the inferior, middle, and upper layers, for example, like shown here, and then basically extract and visualize the feature spaces in depths in different depth levels. Then we are able to translate this kind of ocular dominance and orientation mapping in V1 to any part of the cortex and look into the featural spaces there. So this is just showing you these grids, and there's sample now information on top. For example, these colorful patches here are actually from a real experiment, sampled information in one, but there's another depth sheet in one depth sheet, and there we can now image this information. So I said that we use the so-called Grace sequence, which is a kind of like a spin echo sequence, but we can have multiple slices. And why we prefer this over gradient echo? Actually, we do both. So we compare the results of the classical or standard gradient echo API, but we compare this with the spin echo kind of Grace sequence. And here's the reason. In a recent paper from DiMartino and colleagues, we have kind of compared the two sequences, and we see that the gradient echo signal is actually stronger than the spin echo signal, but the problem is that in the upper layers, when it goes to the superficial, supragranular layers, towards the PL surface, you suddenly get some more activity, but you get a high spread of activity, and you get biases in your sampling. And the reason is that in these upper layers are the draining veins, which basically diffuse the information and kind of deteriorate it. So when we use this kind of Grace sequence, which is more spin echo weighted sequence, then we see basically that we can sample also the upper layers, some supragranular layers, without getting these blurring effects from the draining veins like from the gradient echo sequence. I just tell you that because this comes out from 3-4 studies we have done so far with this comparison, and I will switch back and forth between these two sequences, but I generally would say that the gray sequence has this advantage to be better if you're interested in the upper layers. But both work in these upper layers. There's one disadvantage of the gray sequence. You get only a tiny slab. So you can measure in depths, but you cannot get whole brain slices anymore. So it becomes a bit more like animal studies. So you focus on certain regions and you optimize your 3D volume for that region, and then you scan in this region with high resolution, sub-millimeter resolution, with very good signal, which kind of is largely devoid of these draining brain effects. So now we applied this to, give you one example of how we did this, to a motion experiment. And when I started my fMRI time, we started with very simple experiments where dots like in a star field moved outward, and we compared this to static dots. And that is enough to find the human motion complex, V5, because it responds much stronger when the dots move versus when they are static. And at that time, that was all you could think of to get from fMRI. This was actually done with a 1.5 Tesla scanner, a vision scanner here. And the point now is we want to redo this experiment with the new high field scanning, but with the question in mind whether we can see the features within MT, and not just that MT is active as a motion area, right? So now we want to zoom inside and see the coded features in this area. And what did we do? We presented dots which moved in 8 different directions of motion. These are 2, but here 2 other ones, up, down, left, right. So 8 directions of motion. But we pooled them in 4 axes of motion. Why we did that? If dots move in this direction and they move a few seconds later in that direction, according to Albright, in the monkey, these kind of different directions of motion features and columns in the monkey are neighbored to each other. So if we kind of allow to pool these 2 here, opposing directions of motion, this opposing one, and this opposing one, then we get 4 axes of motion, which presumably have a larger columnar structure, metastructure. That's what we wanted to exploit. It's an aggregation of direction of motion columns into axis of motion columns. And if we do that, then our resolution of 0.8 millimeter was good enough to reveal these structures. You see this here. This is our grids which we put in MT. Here's MT on the slice. And we basically found these kind of color patches for the first time in the human brain, which indicate the axis of motion, which are shown here in different colors, the 4 different axes of motion, which pool always 2 directions of motions. And you see the assigned color. And you basically get maps similar like in V1 for orientation, but this maps now the direction of motion. And that's for the first time visible now in the human brain by using these depth recordings, because you see we get this in different depth levels. Here I showed just 2 from 3 we recorded in 40% and 80% depths. And you see they are similar but not identical. And it's important to see that because we can then separate also the activity in upper and lower layers, which I will show you later in my talk. So that's possible now, and we also get tuning curves. So if we kind of use the mapped features for axis of motion horizontal, vertical, and 2 oblique ones, we get tuning curves which look dramatically similar to the ones you see in macaque monkeys. So it makes a lot of sense that we really seem to here have a resolution which not only shows that an area is active, but that we see the features coded within this area. And that's the idea of this high field, high resolution imaging. So the problem is, of course, if we move forward now, that we basically want to go on now in other brain areas and do the same thing. But the issue is that in V1 and V5, the 2 areas which have been mapped in the visual cortex with columnar resolution, basically we knew what we have to present to probe the right underlying representations, the features, like orientations in V1, like directions of motion in MT. The problem now is, what shall we do to crack the code of areas like LOC, like FFA, like color areas and so on? So some are more obvious than others. Visual word form area we are very interested in. So how do we probe the right features there? That's where my group is working on now, and other people of course too. And that will be done in the future. But I will stop now this part and show you now another important aspect of this kind of mapping, and that is doing more higher level cognitive functions. The point is, we want not map only these features to know these features, but we want to also use them to understand visual cognition. And that is what I want to show you in one example. There are many others, but here's an example of the so-called plate stimuli. And you see that there are two gratings. Here you see the white bars, and here you see the darker gray bars. And they move, one in this direction, one in that direction. And on top of each other, you might see, after some time looking at this, that they fuse to kind of a diamond shape and move upward. It's an ambiguous stimulus which switches in your mind back and forth. Sometimes you see two gratings left-right. Sometimes you see one stimulus going upward. So we can probe now in the brain and ask the question, and this is what we hypothesized, can we expect that if you perceive left-right, that more the horizontal features come boosted, right? And if you see it, the same stimulus up-down, are then the columns for vertical axis boosted? And that was here the question, and we basically did this experiment, and we sampled with our grids, again MT. This is actually the data from the gradient echo EPI study. We could get pretty good maps, but not in the higher layers, but in the middle and lower layers. And that was enough for here, for this experiment, because we are interested not in the layers so much here, but in the populations which underlie this ambiguous percept. Here you see the mapping was not done with dots. It was done with gratings, single gratings, moving in axis of motion directions here. But we could replicate our previous work by finding these kind of columnar maps within MT. But the interesting part is actually, do they overlap? That means we have here the horizontal vertical map within MT. Then we have an induced percept of component, which is this motion in the plate stimulus and ambiguous stimulus, or up and down motion of the same stimulus, but we induce that by some stimulus changes, and then we have the full ambiguous stimulus where nothing is changed. It's just in the mind of the person what they perceive. And if we look at the horizontal vertical columns, they indeed, it's not so well visible, I'm sorry for the colors here, but 66%, so 2/3 of the horizontal versus vertical population are the same voxels consistently across slices and subjects, so that we can say that indeed the horizontal columns are boosted when you perceive in the ambiguous stimulus horizontal motion. And if you perceive vertical motion, the kind of vertical population of mapped axis of motion columns are boosted. And this means nothing more that we can now predict the content of consciousness from the level of cortical columns. This is what this means. And you see this also here if we look at a more fine-grained analysis. So if we move now in, you see this is the slab we have. These are the populations. The blue are for, this is the ambiguous stimulus. Now the subject presses a button and says, I see component. Blue is the component population. Now the subject sees, I see pattern. Red is the pattern population. So you see there is always, when it switches in the subject's brain back and forth, they press a button, and we relate this activity and can really see that it corresponds. And finally, if we look at the individual trials, you don't see this as perfect. An average is very clear, but still you see it as in the best voxels that indeed when the subject sees component motion, you get a boost in a single trial. It goes down, pattern, component up, and the pattern voxels have the opposite behavior. So you find a very nice correlation of component and pattern perception with the horizontal and vertical mapped features. So where does this go? The other aspect I want to show in one example is actually that we not only can use this resolution, as I said before, for tangentially mapping the cortex, but also we can map the cortex in depths. And of course, we do not claim to see all the 6 layers and sublayers with fMRI, but at least we can separate the supragranular layers from the middle layers and the infragranular layers. And that's very important because according to the canonical microcircuit of the cortex, the middle layers get input, bottom-up driven input, whereas the upper layers get feedback input from higher areas. And if we are indeed able to separate this, we are really in the position to at least roughly separate bottom-up from top-down input. And that's not only interesting in itself, it can also be used to test theories about visual attention. Does it really work on upper layers in V1? We can also use it to constrain so-called predictive coding theories, which have specific assumptions, how top-down information influences upper layers or other layers in earlier cortex. And that's why we are so excited about this possibility. Besides the tangential resolution, the depth resolution gives us also these possibilities. And I want to demonstrate this with one example, but this is just to show that we have now expanded the so-called population receptor field technique to multiple layers. So we basically can map and find out without a priori models like a Gaussian. So we fit completely freely the shape of receptor fields in V2 from V1, and actually even vice versa, in different layers. So we get a very high resolution now, layer-dependent estimates of the shape of receptor fields. And the example I want to show you is actually shown here. It's from my colleague Lars Mukli. And actually they had already a nice paper published where they showed with 3 Tesla imaging, if you use a classifier to learn which of 3 scenes someone is seeing, then of course the classifier can do that. This is known in V1. But their trick was to spare out a quadrant in the test phase so that you used only the visual voxels which are not stimulated bottom up, right? So there is no bottom up input. The voxels do not look like activated at all. But nonetheless, machine learning, the population of these voxels from this quadrant could still predict what you have seen. And the only explanation is that this must come from top-down effect, right? And we in the collaboration wanted to find out whether this works at specific layers. So what we did is we repeated that experiment in Minneapolis on 7 Tesla and in Maastricht, and basically found that indeed when the stimuli are shown fully, so that we get from this quadrant a bottom-up stimulus, the classifiers which are now working at different laminae, not laminae is too much, but at coarse representations of laminae in fMRI, and you see that the middle measurements from kind of the middle layers work best when the full stimulus is shown. That means when these V1 patches see this quadrant fully, they can predict what the subject has seen. But the exciting part is when this test stimulus was used for training, the classifiers work best not in the middle and lower ones, but only in the supragranular layers. You see here is the highest performance. When no input is there, the classifiers pick up the information from the upper layers. And that's precisely what is predicted from top-down effects, from predictive coding theory, from expectation theories. So this makes a lot of sense. And this is, again, highly consistent across subjects. So if you see here, other subjects show basically the same finding. You see here, these are almost all subjects have a similar shape of response pattern. So this is the second thing we can do. And to round up, I want to summarize and say that ultra-high field imaging gives us the possibility to extend the macroscopic view, which I showed at the beginning, into the aerial view, zooming into the areas and resolve there tangentially columnar-like structures and in depths laminar-like structures, good enough at least to constrain computational theories of visual processing. And it's best to compare this high field imaging to optical imaging in the animal, because we also get not as good, of course, a resolution like in optical imaging, but some good resolution here, which is better than the standard conventional imaging, at the cost that we lose often whole brain coverage. So these experiments are no longer whole brain coverage. And with that, I want to conclude. I want to just summarize that to better understand brain anatomy and function, multiple levels of brain organization need to be integrated, as I've shown, the view from the beginning, from the pathways to the areas, to retinotopy, to columnar structures. This is all what we try vertically to integrate. And the submillimeter ultra-high field fMRI is an important new tool which helps to bridge the macro and the mesoscopic scales in a functional way and related to structure-functional anatomy. Recent fMRI experiments show that this is possible to map known features, but it's difficult and challenging to go further now beyond V1 and V5 and to map the features in areas where we do not yet know what the features are. This is the real challenge which we and others want to tackle next. But to reveal feature codes in specialized areas at mesoscopic scale has the potential to also shed lights on perception and cognition, like we have seen in the ambiguous plate experiments. Other examples in our and other labs are attention experiments, where we see already that the upper layers in a spatial attention task get modulated by top-down expectations. And finally, to link the feature codes with the possibility to measure roughly laminar profiles helps us to separate bottom-up from top-down processing, which again helps us to understand better how information is coded and especially how areas interact with each other at the mesoscopic scale. Thank you very much.
Thank you, Rainer. Great talk, like always. We have room for one question. Carl, you would like?
From where comes the information for the top-down modulation?
Very good question. Of course, you know, I was not this night, which was strange, you have 12 hours shift like me, but I once woke up in a hotel room and opened my eyes. At that moment, I saw some 2D patch, some black thing here. And it took some moment, suddenly I saw this was a bird sitting on a tree a few meters away. So I saw myself, my own visual system in slow motion. This was an amazing experience. And I think, I tell you that, because I think that when we see in a room, we are constantly full of expectations. We learn and adapt to what we have already in the brain, and this is what I think where the top-down comes from. It's spread in the brain. It's frontal, it's visual, higher level visual. They all together form these expectations. We don't know much where it really comes from in detail, but I think we are now beginning to at least understand a piece of this puzzle, and I think the next step is really to go and try to find concretely which other regions send which concrete information down to the early cortex.
Yeah, thank you, Rainer, again. Thank you.